Hello, I am Dr. Saikat Bhomik, Assistant Professor in Department of Physics, IIT Guwahati. My research group focuses on perovskite nanomaterials, their synthesis, their photophysical study, and different type of applications like energy conversion, detection, and biological applications. Recently, our research group, in collaboration with Professor Chandan Goswami's group in Niger, Bhubaneswar, found that perovskite nanocrystals can be used as a detector for detection of mercury ions inside the cells. So, here actually the ions play a critical role inside the cell for different type of biological functioning. If the excess of ions and deficiency of ions inside the cells that cause serious health issues. We use perovskite nanocrystals that are actually very small, less than 100 nanometer, and it can be synthesized very easily. It can be color tunable from in very visible to infrared region. These perovskite nanocrystals have some special properties like multiphoton absorption. That means if you can excite low energy photons or infrared photons that can give you visible emissions, that is very useful for better imaging quality and even better penetration depth inside the cells for lower scattering inside the cells. The perovskite nanocrystals emission spectra also very narrow. That gives you a better limit of detection of mercury ions inside the cells. The perovskite nanocrystals usually we synthesize by room temperature open atmosphere. However, the perovskite nanocrystals are not so stable in water. So, our research group mostly focus on how we can improve the stability of these nanocrystals. So, we coated these nanocrystals with different type of coatings such that they can stable inside the water for longer period, and we can use them in biological cells. We found that these perovskite nanocrystals can sense mercury ions with very low values in nanomolar. These nanocrystals can go inside the cells because their small is less than 50 nanometer, and we can easily detect these ions inside the cells by simple imaging techniques with help of confocal microscope. Our synthesized perovskite nanoparticles synthesized in our laboratory. You can see these nanoparticles are synthesized from blue to green and red colors, and they are highly luminescent. This is very simple synthesis process. You can see here we can take one solution and mix in another solution to get luminescent green nanoparticles. In the second video, we can also make the blue nanoparticles, and in third one, we can synthesize red emitting nanoparticles from the green nanoparticles. And all these cases, the PL intensity very high, and quantum will reach up to 90%. And you can see this is the size of the nanoparticles taken in TEM. Their particle size less than 50 nanometers. This is the image where you can see with mercury addition the luminescence of the nanoparticle decreases or PL quenches. From this we can say that there is mercury ions inside the solutions. For other ions it is not decreasing. And this is the image where you can see for normal nanocrystals, if they are present inside the biological cells, they are highly luminescent. But if you add mercury inside the cells, the PL intensity decreases. This is how we can understand the presence of mercury ions inside the cells. Thank you.